Now I have the pleasure to introduce the second speaker, Professor Dr. Ola Kamal, in Hospital Central Dolidivans in Spain, Madrid. She will speak about evaluation of intracranial oligodendroglial programmed progenitor cell therapy experimental study.
Assalamu alaikum. My name is Ala Muhammad Fatih Kamal. I'm a physical medicine and rehabilitation. I'm a physical medicine and rehabilitation medical doctor at Hospital Central de Defensa in Madrid, Spain, and I'm also a research collaborator at the neuroscience lab at the Research Institute at Hospital Clínico San Carlos, where I did my residency training earlier on. Today, I'm gonna present you our work titled Evaluation of Intranasal Oligodendroglial Progenitor Cell Therapy for Remyelination in a Multiple Sclerosis Experimental Model. There are no conflicts of interest to disclose. Here is the outline that I'm gonna follow for the presentation. And as many may know, multiple sclerosis is a neurodegenerative disease of the central nervous system that has a very big impact worldwide because of the millions of people that affect, and also because of the disability that causes, especially in young adults. Even though this disease is by definition a disease that produces the myelination, there are no treatments that target specifically myelin restoration. In our lab, we have been researching for years the role of OPCs since they are essential in the process of myelination as a possible therapy to restore myelin restoration in multiple sclerosis. We always faced two main challenges. Those are the availability of the cells and also the way of administration. Anyone here who works with cell therapy will know that it's very complicated to make the cells survive once they are administered, and they have to be administered directly by surgery. To address this problem, in our lab we created a formula with OPCs and biomaterials to ensure the survival of the cells, and at the same time to ensure a non-invasive way of administration or intranasal administration. Our purpose then of this study was to assess whether OPCs administered intranasally could promote myelin restoration in a multiple sclerosis experimental model. Regarding the methodology and the study design, we used 39 Fox-Nude anemic mice, which are a kind of mice that is ideal for xenotransplantation to prevent OPC rejection. Those mice were then exposed to a cuprizone diet for 5 weeks to induce the myelination. And here in this slide, you can watch the 5 experimental groups that we had for our study: control, sham, cuprizone, and 2 other groups that were exposed to cuprizone and afterwards received the OPC therapy in 2 different dosages. We followed a structured timeline. First, we identified the different mice with microchip implantation, and we performed behavioral tests every 10 days to assess functionality. 1 week before sacrifice, we performed MRI scans to assess myelination, and after sacrifice, we did different histological and molecular analyses to assess treatment safety and also to assess the myelination process. I'm going to start with the results of the behavioral test, specifically the open field test. In this test, we place the mice in a box for 5 minutes and we watch the trajectory of the mice recorded by software, and we record different variables in order to assess locomotor activity, exploratory behavior, and anxiety. We have here 4 representative images of each experimental group. On the left side, here is the control group. This is an image expected of the trajectory of a healthy mouse. And we see how after cuprizone exposure, the mice tend to stick on the edges of the box and do less inner crossings. That's because the mice is more anxious and has less exploratory behavior, because the cuprizone, it's typical that it has some cognitive impairment in the mice. After the therapy, we see how the pattern of the mice is partially restored. In these graphs here, we see how in the cuprizone group there is a significant decrease of the distance travel in total. And now I'm going to talk about the walking beat test, where we place the mice on a walking beat and we record the time that the mice need to cross the beam, and also the number of foot slips to assess locomotor activity and balance. In our case, we saw an increase of the number of foot splits in the cuprizone group, and at the same time, interestingly, we saw that in the cuprizone group, the mice transverse the beam faster significantly compared to the other groups, which is contrary as expected, and this can be due to different factors. We also performed the grip strength test to measure the forelimb force of the mice, and we saw how in the dose 1 group, on day 21, there were significant differences compared to the control group. In the mice during cuprizone exposure, before therapy, there was a decrease on the force of the mice as expected. We also performed the inverted screen test to assess fatigue and the marble boring test to assess exploratory activity, anxiety, and hippocampal lesions, but we didn't find any significant differences. That's why I'm not going to explain the results of these 2 tests. I'm going to continue with the MRI T2 images. On the right side, we see a representative image of the control group where the corpus callosum appears as an electrodense structure here. And we see how after cuprizone exposure, the signal of the corpus callosum is attenuated because of the process of the myelination and neural loss, which means that our experimental model is successful. At the same time, after therapy, we see how the signal here is partially restored. We quantified this in an objective way with the software to measure the lesion area, and we saw that in the cuprizone group compared to the control group, there was a significant demyelination that was significantly restored both in those 1 and those 2 groups. Only significant in the those 2 groups. Although there was a remyelination in both groups, but it was only significant in those two groups. Here, the histology results, we saw that the therapy is safe, that does not cause any tumor formations, and at the same time that the cells arrive at the lesioned area. In these images here, the OPCs that are yet alive are highlighted in green, and we see how they are in the hippocampus and also in the corpus callosum, where the myelination process takes mainly place. We also performed Western blot analysis to quantify the proteolipid protein, which is the K protein, in the process of myelination. And we saw how in the cuprizone group, the quantity of myelin decreased, and after therapy, the quantity of myelin was partially restored, being the recovery greater in those two treatments. Those results were further supported by the immunofluorescence of PLP for the same protein. And we have here a representative image in a healthy mice of a normal quantity of PLP. And we see how after cuprizone exposure, this signal is lost, and how is it partially restored, both with the dose 1 and dose 2 group. In the right side of the slide, we have the statistical analysis shown in a graph, and we see how indeed after cuprizone exposure there is a significant demyelination exposed to the control group that is significantly restored after the dose 1 and dose 2 therapy, being more significant in the dose 2 therapy. And to end with my results, we also performed transmission electron microscopy to assess myelination. And in these images, you can see axons of the neurons cut transversally, and the myelin here is shown as an electrodense structure surrounding the axon. This dark structure, more electrodense, is the myelin area of the axon. And we see how after cuprizone exposure, this signal is again lost, and the axons have thinner myelin around, and how is it partially recovered after therapy. This is again objectively quantified with the G ratio, and we see in this graph how, indeed, after cuprizone exposure, there is a significant demyelination process that is afterwards partially restored with both dose 1 and dose 2 therapies. To conclude, intranasal OPC therapy is safe and promotes remyelination in an experimental model of multiple sclerosis. The correlation between the myelination and remyelination with functional behavioral tests remains limited, and it is essential to consider this study's limitation when designing future trials to optimize dosing, assess the benefits of this therapy, and evaluate its functional impact in a way that could be meaningfully translated to humans. Thank you for your attention. This research is the first step toward the non-invasive regenerative therapy for multiple sclerosis, and we hope to move closer to clinical application and ultimately improve patients' lives. This is the team that I have in Spain working with me in the research lab. I want to highlight the multidisciplinary effort with many kinds of professionals, neurologists, PMRs, biotechnologists, psychologists, engineers that have made this project possible, and also the results of this project.